The plan for today is to finish up the remaining few animal lineages that we have not yet touched on. We'll be going through 3 different phyla, the arthropods, the echinoderms, and the chordates. Obviously, we'll be giving them very short shrift. We'll just be spending a few minutes to try to give you at least a touch of the flavor of these important different groups. And I wanted to, well, let me also first make a few reminders and announcements. Of course, test 2 is on Friday. Just bring your photo ID and a pencil, please. I have updated the Not Responsible For document in large part because there's been a large amount of questions on Piazza, which I hope you're all visiting. I think it's a really useful way for you to get information about the class from each other and from the other instructors, the TAs and I. There's a lot of questions about exactly what kinds of information you're responsible for on the different lineages. I'll try to make that as clear as possible, but I thought I would say something about if you consider how we actually learn and process things effectively, our brains are kind of wired for stories, not bullet points. And so if you're forcing yourself to study by looking at lists of bullet points, that really is not only is it not enjoyable, but it is also not a very effective way to process and learn information. You'll do much better if you actually think about information as a narrative. And that is one of the reasons why, as we're walking through the different animal groups, that we'll go through scenarios or stories about some of the examples to help you learn and understand the differences between the different animals. Is there some memorization of factual information? Absolutely. When we go through phylogenetic trees or consider the names of different taxonomic groups or key characteristics, but much of the information, I think, can be better understood as a narrative. Let's see. The peer tutors, the BioSci peer tutors, have asked me to remind you that there's a review session tomorrow evening from 6 to 8 in SSLH 100. For more information, you can go to the BioSci peer tutor website. I was also asked to make a couple of other announcements, so here they are. I got an email from a student who is involved in a public health research program that studies what UC Irvine undergraduates do for stress relief. So if you're not stressed, then you would not be a good participant. If in fact you do occasionally feel stressed, then perhaps you would like to participate in this study. If you're interested, all this information is available on the Clubs and Other Opportunities tab. The Biological Sciences Student Council wanted me to let you know that tonight is the Winter Gala. It is right outside here. It's between BioSci 3 and McGaw, and it's from 5:30 to 8:30, and it looks like they will have food as well. So that is tonight. And if you're also free tomorrow night, the Biological Sciences Student Council would like you to know that this is an opportunity for you to have an opportunity to chat about specialized majors with some counselors and upperclassmen who will be available to answer questions. Specialized majors meaning things like the genetics major or the neuroscience major. So that's an opportunity, and they'll be providing pizza. Okay, that's it for announcements. Let's go ahead and dive into our first of 3 phyla that we'll zoom through today, and that is phylum Arthropoda, which is the arthropods. And I hope by this point you are familiar with and used to the different words that are used to describe the actual name of the taxon, such as Arthropoda, versus the name given to the organisms which are in that taxonomic group. So an example would be the difference between arthropods and arthropoda. Arthropoda is the name of the taxonomic unit, and individuals within that, within phylum Arthropoda are called arthropods. So that's why those are different words. For almost all of the groups of organisms that we've been considering, we have not burdened you with identification of the actual taxonomic level, such as phylum. There are some exceptions to that. When we went through the domains, they were identified as domains. And as we've been going through the animals, we are identifying some of the groups as phyla. But other than that, we haven't been burdening you with that. But for all the different names of taxonomic groups, then they do actually correspond to taxonomic units. We just aren't identifying them. So what can we say about the arthropods. Well, this is the largest animal phylum, so it's shocking how little time we're actually going to spend on this. Breaks my heart. It probably doesn't actually break yours. Phew. All right, what a relief. But over a million different species of arthropods have been named, and these are fairly familiar creatures, I hope, to many of you. What are the characteristics of the arthropods? Well, let's think about what is a more inclusive group of animals within which phylum Arthropoda resides. Remember that with the nematodes, the arthropods are within the ecdysozoans, meaning that they have a hard covering or cuticle. Now, there are consequences to being covered by something hard and stiff. Okay, with the nematodes, we saw it was a relatively simple body design and relatively limited movement. In arthropods, there was a different strategy, a different functional design, namely that the hard exoskeleton has a number of places where there is articulation, where it is less hard or a little bit flexible. The cuticle is a bit thinner. And so this then makes areas where there could be joints. So if you think about how this works, something that is perhaps more familiar, because of course an arthropod has a so-called exoskeleton, a skeleton mechanical support on the outside of the body, the reverse of us, right? We've got our hard skeletal parts, our bones on the inside, and we're squishy on the outside. So an arthropod is sort of the same, is the reverse of that, right? Hard on the outside, soft and squishy on the inside. And if you're hard on the outside, then one of the problems with being hard and stiff all over the outside of your body is you then can't move if it's completely rigid. And so in a suit of armor, for example, then how one is able to move if one is in fact wearing a suit of armor is that there are articulating areas or joints between the different rigid plates where movement is possible. And that's exactly what you see in the arthropods, and you can see them as lines when you look at the outside of the creature. So the characteristics then of the arthropods is that they are in parts. Their bodies are segmented from the anterior to the posterior, and any appendages are jointed, allowing for articulation, just as the body as a whole is also jointed. What we're actually looking at right here is an extinct group of arthropods, an example of an extinct group of arthropods called the trilobites. These are known only from the fossil record, and they are representative of what the very first arthropods were probably like, meaning that there was much less variation between segments in an anterior-posterior direction. Even though you might not be familiar with trilobites, it probably makes intuitive sense to you that as you look at this picture, the head is up towards the top of the screen here, and the posterior is down below. But you can see there is some differentiation between the anterior and the posterior areas, but many of the segments are very similar to each other. This is characteristic of some arthropod groups, but certainly the earliest arthropods were more like this. But the arthropods that are still running around on the planet, meaning the extant arthropods, and I think I've complained before about this particular adjective, extant. One has to be very careful when one uses this adjective during lecturing, because it sure sounds a lot like extinct, but it actually means the opposite. Okay, so the extant arthropods are the ones that are not, in fact, extinct, and they look, many of them look much more like the crustacean or lobster that is shown in front of you right now, where there is even more differentiation from the head end down to the tail end as shown here, but also tremendous specialization in the appendages, these jointed appendages that are the hallmark of the arthropod body plan. So up at the front here, we've got some appendages that are sensory, the antennae here, the two pairs of antennae, and then we also have pinching claws, we have walking legs, and then we have swimming appendages. These are all modified appendages, modified in different ways for different functions. They're clearly distinct from each other. So that is what is meant by appendage specialization. And one sees that as an evolutionary trend in the arthropods as a whole. So the arthropods include such familiar groups as the spiders and the crustaceans and the insects and the centipedes and the millipedes. And do you have to know all the characteristics for all of these groups? Absolutely not. But I would certainly hope that if you heard about a spider, then you would know that this was an arthropod. You would be able to know what phylum of animals you could place such a creature into. But we will consider a couple of characteristics of the insects, and this is such a huge and diverse group, and there are more named insects than all other forms of life combined. So it's rather heartbreaking that this is in fact the only slide of the insects, information about the insects that you will know. So what are some distinguishing that you will be exposed to in today's lecture? And what are some of the key characteristics of the insects? Well, they live almost everywhere except marine environments. So this is seen as really quite curious. There are another arthropod group that certainly dominates the marine scene, and that is the crustaceans. But insects, although they're found in virtually every terrestrial habitat and freshwater habitats, very few of them are ever found in a marine ecosystem. You can also tell an insect because it has 6 legs. That is another characteristic. And most insects have wings and can fly. You're probably familiar with this, flies and bees and so on. And you have learned about a couple of examples of insects earlier in this course. For example, the importance of bees as pollinators. Bees are a very speciose group of insects. Others are carriers of disease. There are a lot of different examples of that, where the mosquitoes are among the most infamous and important disease-transmitting insects, and then various other insects are pests of crops. So sadly, we're going to go ahead and leave the insects, but I do hope that when you see words that correspond to groups of organisms that you have been learning about, that you're able to look at such words and have a feel for, is this a more inclusive group or a less inclusive group? How do these relate to each other? In fact, maybe a good way of thinking, especially if you're getting the different groups confused, would be to take a piece of paper, write down the names, cut them all out, and try to arrange them in order of which are more inclusive, which are less inclusive, and which are at the same level. So for example, the words that are in front of you right now, we've got a couple of examples of phyla. This is a phylum, Chordata, and this also corresponds to another phylum. Because these are the same taxonomic level, then neither one could be a subset of the other, right? They're at the same taxonomic level, and these are also 2 groups that are at the same taxonomic level, the deuterostomes and protostomes, although we've not identified the actual taxonomic level. Okay? So as you look at this, then hopefully you can think of what is a subset of what. So let's do this as a clicker question to give you a little practice in thinking about this. So we just chatted extremely briefly about the insects, and so I'm wondering if you are able to identify the correct sequence of enclosing taxa, where these are different taxonomic groups, and you should be able to identify the most inclusive, meaning including a larger, larger set, to least inclusive, meaning a subset within a larger set. So insects then is the least inclusive. So to interpret A, for example, you would say, you would pick this one if insects are within deuterostomes, which is within eukaryotes, which is within animals. Okay? What groups are within others? If you're not quite sure, great time to meet your neighbor. You know, hello, my name is, I have no clue, what do you think? I'll give you 30 seconds or so to think about this, and then I'll check back in with you. Okay, is everybody clicked in? The answer counts, by the way, so you'll get more credit on this if you pick the correct answer. So, all right, let's, hopefully you all got this right. Let's find out. Fantastic. Almost 90% of you got this right. Nice job. And you might have thought, you know, well, you know, not all of these have, say, the arthropods in here, right? Of course, not all of the intervening taxonomic subdivisions are actually shown here, but this might give you an idea of how to practice testing yourself on your knowledge about these groups. But it is time to now move on to the next two phyla, both of which are deuterostomes. So hopefully as we navigate our way through the hierarchical classification system for animals, you're thinking of a visual of a phylogenetic tree in your head. I think that helps to keep things straight. Again, here is a very simplified version of the animal phyla. So we have been talking about the protostomes, right, which are not identified explicitly on here. So I'll go ahead and put that on for you, right? So the Lophotrochozoans and the Ecdysozoans, both of which make up the protostomes, right? And the protostomes and deuterostomes are sister groups, right? Okay, so we finished up talking about the protostomes, and now we're going to turn our attention to the deuterostomes, just focusing on two major phyla. And again, we're just going to hit the high points, and this will also help you, I hope, navigate through the information in your textbook as you're reviewing the different groups. If I don't mention a group, especially if it's something they're not responsible for, you are absolutely not responsible for knowing the details of those groups, okay? So, starting with the echinoderms, and this is really a close relative, so I think it's a bit shocking how clearly distinct the body design is from our members of our own phylum. So various representatives are shown here in picture form. So examples are sea stars and brittle stars and sea urchins and sea cucumbers and sand dollars, all of which might be familiar to you if you go to the water's edge or in shallow water in the seas around here or really around anywhere, because these are all marine. This is an unusual phylum in that every single representative of this phylum is only found in a marine ecosystem, meaning you're not going to find them in a terrestrial ecosystem and you're not going to find them in fresh water. They tend to be fairly slow moving, as you know if you're familiar with any of these creatures. Do you need to know all the gory details about all these different subgroups? No, I'll introduce you to a couple of points about these, but you're not responsible for knowing the taxonomic classification within the echinoderms. Another unusual and a noteworthy thing about the echinoderms is the unusual kind of symmetry that they have. It's a form of symmetry, but specifically it's a five-fold symmetry, which is easiest, I think, to see in the sea stars or starfish, where there are 5 legs or sometimes 5 arms, or sometimes multiples of 5. And so this is considered a form of radial symmetry because one can have a plane of symmetry that rotates around an axis, and there are multiple planes of symmetry as one rotates that plane. So that's why it's considered a form of radial symmetry. This 5-fold symmetry is more obvious for some of these, or for some examples within this phylum, such as the sea stars or the brittle stars. There's an example of a brittle star. It's less obvious in the sea urchins or the sea cucumbers, although if you look very carefully at the anatomy, you can often see details of 5-fold symmetry. In sand dollars, it's a little more obvious. I don't know if you can see. I'll just sort of outline the little characteristic design as typical on the sand dollars, also showing their 5-fold symmetry. Very unusual. No other major group has this kind of 5-fold symmetry. And if you've ever handled an echinoderm, which can happen, for example, if you go to a marine lab or a museum that has living specimens that allows you to handle them, then they feel very hard and stiff, and what you're feeling are the hard calcareous plates, calcareous meaning made of calcium salts, primarily calcium carbonate, that underlie a very thin epidermis, so they feel very hard. Another very unusual structure that one sees in echinoderms is a so-called water vascular system that penetrates the entire body and extends to the outside of the organism through structures called tube feet, which are used in locomotion. And we'll look at a video in just a minute so you can see how these structures are used in locomotion. This is, again, something that is peculiar to the echinoderms. And the only other thing worth noting, perhaps, is that these echinoderms do reproduce via sexual reproduction. They are all in seawater, and they reproduce via external fertilization, meaning that the males release the sperm into the surrounding seawater, and an egg is fertilized when the sperm reaches the egg, which can also be released into the seawater or retained by the female, depending on what kind of echinoderm it is. So this is exactly the same text. I just shrunk it down and put it to the left in case you want, or help you remember the major points about an echinoderm. I'll make it a little darker so you can see the video here. This is just a very short video showing 2 feet in action. So this is just a single arm of a starfish. You're looking at some sort of side view. When you look at the outside of a starfish, you can see a special structure through which connects the water vascular system with the surrounding seawater. But as mentioned, these are very slow-moving organisms. This is in real time. Looks like this individual is righting itself. And so as it moves, and all these little tiny hydraulic tubes here, the individual tube feet then attach and are released and attach and are released. So this is really quite slow movements in these interesting creatures. I'll show you a video of another example of of an echinoderm. We're going to look at sea urchins. They're a different, interesting example because of their very different diets. Many sea urchins are herbivores. Here is an underwater picture of a particular kind that are bright purple. This is time-lapse. These are very slow-moving organisms, and sure enough, they are hungry, as the text says. These voracious herbivores chow down then on the algae, and they're very important consumers of the algae in many of the marine ecosystems, as we'll see later when we go through some examples. Yeah, it's chewing on it. That's the sort of the mouth here, right? It's chewing on the the algae. I think you just totally decimate the seaweed. So the sea stars are often carnivorous and can even open bivalves and eat the insides, but sea urchins are more herbivorous. Give you a little idea of some representatives. I won't take time to go through any of the other representatives, nor are you responsible for the details about their lives, except what I have said, and so hopefully that gave you a little bit of a narrative so that you have a feel for the major defining characteristics of echinoderms. And now, onto our final phylum, Chordata, our own phylum. There are, sorry, the mic volume is jumping around. There are 4 major defining characteristics of chordates, and they're listed and color-coded in the legend that accompanies figure 35.8, which is shown now to you in part with just one representative group of the chordates to walk through the different structures. So what are these 4 key characteristics of all chordates? One is gill slits in a pharynx, that's shown here in yellow to orient you with this particular organism that we're looking at. The anterior is to the left, so this is the head end in this direction, and this is posterior in this direction. You're looking at the side of the organism. And so this structure here is the pharynx, and there are slits in the pharynx. And the significance of pharyngeal gill slits is that this then makes a pharynx an organ that can be used for filter feeding, as is being done by this organism. So if we look at the water flow for this particular creature, which is an example of the group called the lancelets or the cephalochordates, you can see water entering the mouth here, passing through slits in the pharynx, then leaving the body. So this organism is making its living filter feeding, pulling particulates out of the water. Right? So you immediately know if you see something filter feeding, it is an aquatic organism. Moving on to characteristic number 2, dorsal hollow nerve cores. So this is the back of the organism here, the dorsal surface, and so the blue structure here is the dorsal nerve cord. This is something that is a bit more familiar to us. The next structure is the notochord. This is is labeled in pink and is shown right below the nerve cord. Now here is where students find these two structures very confusing because they both have the word cord in them, right? Nerve cord, notochord, and they also both start with an N, okay? But they're completely different. The nerve cord is part of the nervous system, so this is made of nerve cells, right? So what do nerve cells do? They send information, they transmit signals, information flow is the whole, using neurons is what nervous tissue is doing. The notochord is just simply a skeletal element. So this is mechanical support. So this is just a stiff rod. This is the skeleton of this particular organism, skeletal support, the notochord. So one is mechanical and one is transmission of information, so completely different structures. And then the final item here in our list of 4 is the muscular post-anal tail. What does post-anal mean? Post-anal means posterior to the anus, which means we have to first find that part of the anatomy. And so that means we have to find the digestive system. It is actually found here. Here's the mouth at the front end. And then this is the anus, not where the water is coming out. So the water is not flowing along the digestive system. This is the digestive system shown right here that ends right there at this particular point. So this you can tell from the color coding as well. So in the posterior area of the body that is further in the posterior direction, then there is a muscular tail. So as you're considering these 4 characteristics and considering your own personal body design, then it should be obvious to you that only one of the 4 items in this list is something that you yourself possess, at least at this particular point in time, right? And that is a dorsal hollow nerve cord, right? We don't have pharyngeal gill slits, at least not right now, nor do we have a notochord, nor do we have a muscular post-anal tail. So why is it then that we are considered chordates? It's because these characteristics show up in at least one part of the overall life history. So now let's consider figure 35-8 from your book in its entirety. And this then allows me also to identify that the chordates as a whole are divided into 3 groups. 2 of these groups are considered invertebrates, and then the 3rd group is the vertebrates. So the first group here, the cephalochordates or lancelets, we just spent some time on. The second group is the urochordates or tunicates, and I'll say a few words about them in just a minute. And then here's group number 3, the vertebrates. And for many of the vertebrates, our own group included, then these 4 items are only found during early development in the embryo. So certainly in our embryonic stages, we did in fact possess these different characteristics. And so this these shared characteristics then is yet another example of how developmental biologists were able to use characters that are shown very early in life, in the life cycle, to identify the relationships between otherwise very disparate organisms. So, so I do expect you to, as we go through the chordates, we'll hardly touch on any groups at all, and I'll be very clear at identifying them. I do expect you to know that chordates are divided into these 3 groups. The first 2 are really small. We'll blitz through them right now, and then the 3rd one we'll spend a little time. We are certainly not going through all the vertebrate groups, as you'll see as we go through the vertebrates. But let's go ahead and start with the cephalochordates. This is not a picture from your book. I took this from elsewhere because it's a little easier to understand the typical life of a cephalochordate, or its common name, a lancelet, if one sees it in its natural habitat, and which is buried in, typically in sort of a sandy surface, such as is shown here. This small, somewhat fishy-looking organism then has its anterior end sticking out into the water, as shown here. Remember, these organisms are filter feeding, so they're drawing water into the mouth, passing them through the pharynx, through the slits, capturing particles, and then the water exits, and that's how they make their living. The name lancelets come from their blade-like shape. So a lance is a kind of sharp blade, and so that's where this name comes from. Okay? And I am not actually saying anything else about the lancelets except to point out that the very first chordates are thought to have have been quite similar in many respects to today's lancelets. The second group, the Urochordates, are similar, at least in the larval form. This is a picture of a tunicate larva in front of you right now. Also a marine suspension or filter feeder. Also draws water through the mouth, passes it through the slits of the pharynx, filters out some particles. That's how these organisms make their living. They look a little tadpole-like. They are absolutely not tadpoles, but they do have a tail and then a somewhat more bulbous anterior end. They're very active. They swim around. This is in huge contrast with what the adults look like, which is shown in this particular picture here, because the adults are sessile, meaning they're attached. In fact, they remind many students of sponges because they are sessile attached suspension feeders. And at this stage in their lives, they've lost virtually every indication of their chordate history or chordate relationships, because the notochord is gone. The postnatal tail, gone. The dorsal cord, gone. These individuals have been reduced to an extremely simple morphology in the adult stage. They still have the slits in the pharynx, and they still make their living filter feeding, so they still process water. They're sometimes also referred to as sea squirts because they occasionally squirt out water. Okay, so that's two of the groups, and this going through this, taking time to go through this, hopefully will help you understand the evolutionary history of the chordates and cementing the idea that the very first chordates were presumably filter feeders or suspension feeders in a marine environment. This is figure 3512 in front of you now, which shows a phylogenetic tree for the chordates. And so we are absolutely not going through all of these different groups, but I would be very remiss if I didn't at least take a few seconds to cover some of the major evolutionary innovations that occurred within diversification of the vertebrate lineage. So let's take a little time to orient ourselves here, and then we'll just pick a very small number of groups for a little additional information here. So this whole group then is the chordates, right? We've already covered the cephalochordates and the urochordates. Check, check. And now the rest of these, the whole rest of the phylogenetic tree is now the vertebrates. So you absolutely are not responsible for any of these terms, but if we sort of ignore then the names of the different taxonomic groups, then as we're considering the different groups here, we can at least consider some of the evolutionary innovations that did occur during the evolution of this group. So if we start here with the vertebrates, then it should not surprise you to see that an important synapomorphy for the vertebrates are vertebrae, right? Hence the name. So these are important additional mechanical skeletal elements in addition to the notochord that the non-vertebrates had as their only skeletal element. So the vertebrae at first were not bony. In fact, that is a later an evolutionary change in some of the lineages. So the very first skeletons then were cartilaginous or made of other kinds of materials and were not bony. That was a later later innovation. So what else can we see here? Jaws and lungs, lobed fins. We are going to pull out this particular innovation, though, for some consideration, and that is the evolution of the amniotic egg. This is a very significant and noteworthy modification of some of the lineages because of the way it changed the possible habitats that individuals that did have an amniotic egg could inhabit. So those individuals that have an amniotic egg are called, mnemonically enough, the amniota, and there are only two groups within the amniota, the mammals and the reptiles. So let's zoom in then on that little part of the figure so you can see this a little more easily. So we're considering the evolution of the amniotic egg and the consequences for this structure in the lives of those organisms that had it. So in the major issue then with the mammals and the reptiles is now the presence of an amniotic egg meant less reliance on an aquatic environment. So do reduce dependence on an aquatic environment, meaning that these organisms could then invade the terrestrial environment. This is certainly less so for the amphibians, which as a group are much more dependent on water. So what is it about the amniotic egg that allowed invasion of the terrestrial environment? This is a figure that shows the parts of an amniotic egg. This is figure 35-17 in front of you right now, and the shell is a little hard to see here. So this is a diagram of the different parts, and there isn't always a shell, but sometimes that's this structure right here is a little pale. So there's the shell. As you look at the different parts of an amniotic egg, then you can see the embryo developing here right in the middle. And then there's a whole series of complicated membranes that subdivide the environment into different parts. But what this does is it provides basically an aquatic environment for the embryo that is independent of the external environment. So this then is what this particular form of an egg then made inhabiting a terrestrial environment then more possible. All these different membranes have different names. You are absolutely not responsible for knowing all the different names and who does what. You will see these names again in just a minute, but I think it is interesting to note that these different membrane-bound containers have really quite different functions, right? You've got the yolk sac containing the nutrients, and you have a different compartment for wastes and so on, and then a different one containing the embryo. So I think that's all that I want to say about that. So within the amniotes then, we've got the mammals and the reptiles, right? We're not going to say anything more about the reptiles. We're now turning our attention to the mammals, our own group, and we're going to say virtually nothing about the mammals except to note the important synapomorphies here for the mammals. Mammals are a monophyletic group. And lactation and also fur. What is lactation? Lactation is just simply the production of milk for the young in structures called mammary glands. That's where the name comes from for the group, mammals or mammalia. So fur or hair is another synapomorphy for the mammals. Other things that are noteworthy to point out about the mammals is that they are considered endotherms. This just simply means that they characteristically have a body temperature that is elevated and regulated above the surrounding environment. We are examples of that. Our body temperatures are virtually always elevated relative to our surrounding environment, and the source of the heat is internal. We're generating our own heat. That's where this name comes from, endothermy. Mammals in general tend to have fairly high metabolic rates relative to the other non-mammalian but closely related vertebrates, fairly large brains for body size, and then differentiated teeth, which is important for those individuals that study differences between different mammals. We are not going to say anything about more about that. So the only thing as mammals as a whole, we will consider that mammals as a whole are divided into three lineages, at least the ones that are still extant, and that's shown in figure 35-22, which is in front of you right now. So here we are, the mammals. Of these lineages, two are really quite small. The monotremes and the marsupials are very small. We'll go through these groups rapidly and just connect this with the reproductive physiology or reproductive attributes of these different groups. So let's go ahead and dive right on in here. The first group, the monotremes, tiny little group. It's just the echidnas and the platypus. That's it. Here's a picture of an echidna. They're only found in Australia. That's it. One of the extraordinary things about the monotremes is that these are egg-laying mammals, and this is thought to be an ancestral condition for the mammals as a whole. So this is the only group out of the three that still lays eggs. So if you didn't know that any mammals at all laid eggs, well, here's the example for you. I think that's all I was going to say about monotremes. Let's move on to the marsupials, which in terms of their young, production of young, are sort of intermediate between the monotremes and the other group, the eutherians. And so marsupials, which is also a relatively small group, but it includes the kangaroos, the opossums, and the koalas. Marsupials do not lay eggs. They do give birth to live young, but at a very, very early stage in development. So when the young are born, they're often even described as still embryonic when they're born, and they're completely dependent on the mother and access to the mother's milk, and so they complete development while they're nursing, while they're getting milk from the mammary glands. You're familiar with the pouches in kangaroos. That is an area where the young are while they're still developing. So the last group, the eutherians, when the young of the eutherians are born, and this is the largest group, virtually every mammal you could think of is in this group, with the exceptions that we just covered. So primates and rodents, dogs, cats, zebras, bats. Everything else is in this final group, and young in the eutherians are much more developed when they're young. When they're born, they still nurse from the mother and still get milk from the mother, but they are born at a much more developed stage, and so this corresponds then to longer pregnancies in the mother. And then while they're within the mother, then they are receiving nourishment from a structure called the placenta. This is shown diagrammatically in figure 35-18, which is in front of you right now. So just to orient you, this pink structure surrounding the developing embryo here is supposed to represent the organ, the uterus. Here's the developing embryo. The placenta is an unusual organ that is where the mother's blood is brought into close proximity with the fetal blood, or the developing fetus blood. And you can see a number of different membranes, and if you remember what the names are, I'm not wholly responsible, so I'm guessing you may not have even noticed what the names were, but there's a whole set of different membranes that have the same name because they're directly homologous to the structures that one sees in all amniote eggs, as shown here. So let's turn our attention now to the primates. You are absolutely not responsible for the relationships between the different primate groups. Here are humans right here. You should certainly have an idea of what primates are, and that this includes gorillas and orangutans and chimpanzees and humans and so on. But here's a closer view of humans with the closest relatives. We're going to focus on the hominins. This is figure 35-37 in front of you right now, and what I first want to point out is this is not a phylogenetic tree. What this is a representation of the known appearances of the different hominins in the fossil record. So the present time is at the far right, and then going earlier, you go to the left. Homo sapiens, our species, is the only species out of all of these that is still on the planet. And you can see there are various other species within our same genus Homo, as well as different genera that are close relatives to us. You are absolutely not responsible for learning all of these different ones, but I would hope that you would take some time to look at this figure and think about the implications of this figure, such as Homo sapiens are only known from about the past 200,000 years or so, and that Homo sapiens shared the planet with a couple of other species of Homo for some time. So how to read this graph is that each rectangle shows the first to the last appearance. So if we then, so this left boundary on Homo sapiens then shows when we first were identified on the planet, and so you can see that we did overlap a tiny bit with Homo erectus and more so with some of these other groups. And some of the other groups went extinct actually fairly recently. Now there has been an evaluation of the relatedness between the different human populations living today. This is figure 3539 in front of you right now. And because this is just one species, then these different branches are not are different populations. And so one can look at the relatedness between the different groups from different parts of the Earth, from different continents, and get some, make some inferences about the early movements of humans on the planet. And that is shown diagrammatically for you in figure 3540, which is in front of you right now. It is pretty clear that Homo sapiens originated in Africa and then spread throughout the world. And one can get dates from the fossil record. It is thought that there were a variety of different waves. And what has been interesting more recently is that, to go back to the, because our other close relatives have disappeared from the planet, there's interest in knowing, well, did Homo sapiens displace the other species? Did we interbreed with the other species, meaning that perhaps they weren't really other true species? Scientists have been able to pull some DNA out of some different fossils from both the Neanderthal species, this one right here, and also Homo erectus, this species right here, both of which did overlap in time on the planet with Homo sapiens. And some populations of humans actually have clearly identified DNA that is probably Neanderthal in origin, and their populations with Homo erectus. Which populations and how would this occur? My remaining 20 seconds here. None of the African populations have evidence of Neanderthal or Homo erectus DNA. So it's thought that as Homo sapiens migrated out of Africa, there was some interbreeding with other populations as they spread, because this is just shown in some of the lineages that then spread other areas of the, on the continents. Okay, I'm now out of time. I'll see you on Friday or possibly Thursday in office hours.